Okay. Okay, so please don't be intimidated by the amount of slides. Remember, half of these slides are just the answers to the slides before them, which we're not going to have to stay on, okay? So biotechnology, okay, go. This is a huge topic. We're going to boil it down to two things. Electrophoresis, which we'll do today, bacterial transformation, which we'll do next Tuesday, okay? Okay, so DNA electrophoresis. In this process, a visual image of a DNA sample can be made by running its cut fragments across a special gel and analyzing the pattern created. This technique has many uses. You can make a fingerprint for crime scene investigations. You can compare DNA samples for paternity testing if you have the child's and the mother's and you want to know who the father was. Comparing DNA of different alleles to detect mutations. Analyzing viral DNA and sequencing DNA. These are all things that you can do. Now realize what you can't do with gel electrophoresis, and everybody gets this confused. You can't take a DNA sample and make lots and lots of copies of it. That's not what this does. That's PCR, preliminary chain reaction, which you might have heard about, okay? Which we aren't going to study. So you can't augment a sample with just DNA electrophoresis. You can only analyze what you've got. You also can't engineer anything with this. This isn't engineering. This is just analysis of a particular DNA sample. People get that confused too. So making the visual image. This technique uses a gel. And since you guys are here, maybe I'll carry one around for you so you can feel it. It's kind of a cool thing. As a molecular sieve to separate nucleic acids or proteins by size. Generally, it's by size. The longer fragments don't travel as far as the shorter fragments. And you use an electrical current so that the molecules move through the gel and they get sorted into bands. Okay. All right, restriction enzymes. Very, very important. They're like a set of molecular scissors. That's what they are. They're like scissors. And what they do is they cut DNA into small fragments. And that's what you have to do before you do gel electrophoresis, because now you have a sample with different size fragments that are going to go different distances, and that's what creates the scattered bands. Where do they come from? So this is actually kind of interesting. Why would you have something that cuts DNA up? That sounds like a dangerous thing, doesn't it? We wouldn't want to have that in our cells. That might be not a good thing. But it turns out they come from bacteria. Bacteria have been waging a really long time war with viruses. Viruses have been attacking bacteria ever since bacteria showed up in the bogs and swamps of the planet. Viruses were the first predators of bacteria, really. And viruses, remember, a virus injects its DNA in order to infect a cell. So if you have viral DNA in a bacteria cell, it's handy to have a set of scissors that can go and just attack the viral DNA and leave your own DNA alone. So it actually was adaptive for them. But now scientists were able to take those restriction enzymes out of bacteria and use them as a tool to do this DNA analysis. So there's a video here that I want to quickly share with you. I think you guys will go there, but you guys won't. So I'm going to share this little restriction enzyme video because it really describes very well how these guys work. So I'm just going to turn my sound off, and I just want you to. I didn't share audio. Okay, hold on a second. I'm going to share the audio first. There.
In this video, I will be discussing restriction endonucleases. Restriction endonucleases, also called restriction enzymes, are enzymes that cleave DNA at specific sequences. These enzymes are particularly valuable in a lab setting because they allow scientists to cut, isolate, and manipulate DNA segments of interest. Restriction enzymes are naturally found in bacterial cells. These cells use their restriction enzymes to cut apart DNA that has been injected into them by viruses. Cutting up the viral DNA prevents the virus from successfully reproducing within the bacterial cell. The key is for the restriction enzymes in the bacterial cell to recognize sites that are only found in viral DNA and not found in the bacterial DNA. The bacteria wouldn't want its own DNA to be cleaved by restriction enzymes. So as I've said, the restriction enzymes can only recognize and cleave DNA at specific sites. This site is called a restriction site, which is often 4 to 12 bases in length. For example, the restriction enzyme EcoR1 recognizes and cuts DNA at this site. It will scan the DNA, and when it finds the site GAATTC, it will cleave it like this. You'll notice that this cut does not go straight through the two strands. Instead, it creates what is called sticky ends, where these 4 bases are left without their complementary partner. Having sticky ends makes it easy to attach other DNA molecules that have been cut by the same restriction enzyme together. So we can see here that this molecule has 2 restriction sites. It will get cleaved in these sites by the restriction enzyme, and now we have 3 separate molecules of DNA. This second molecule of DNA has had a substitution mutation in the second restriction site. This means that the restriction enzyme would not cleave this molecule here. Instead, it would only cleave here, creating 2 separate fragments of DNA.
All right, so this is a really typical AP Bioessay question right here, all right? This is the kind of thing where they'll show you a piece of DNA and then, oh, see, there's a mutation right here. And then they'll say you're using the EcoR1 restriction enzyme, and this is where it's supposed to cut. And then they'll ask you, how many cuts is it going to make? And how many fragments would be created in each case, before and after the mutation? So that's a really typical thing that you might get in an FRQ, or even in a multiple choice question.
So in a lab setting, what do you do after you've cleaved a DNA molecule? You can separate these DNA molecules using gel electrophoresis, and then after separating them, isolate a piece of interest. You can take this DNA piece of interest that you've isolated and insert it into a plasmid that's been cut by the same restriction enzyme, and then you can insert this plasmid into a cell to transform bacterial cells.
All right, that takes us more to next week, bacterial transformation, but I wanted to make sure that you understood restriction enzymes, and that's really where I'm going to stop that video today. Okay, so now let's go back to our presentation. Here we go, back to the presentation, and let's see if you can answer these next few questions. There we go. See what you can do with that. Let's see how you're doing here. That's it. B is correct. Beautiful. An enzyme comes from bacteria, cuts DNA at a specific base sequence. Beautiful. All right. And what is a restriction site? The enzyme are the scissors. The restriction site could be described as okay, let's see. A specific nucleotide base sequence where a restriction enzyme cuts. That is it. Yes. Okay. The fragment is not the same. The fragments created by the cuts are not the same as the actual base sequence where the cut is made, right? So there's the restriction enzyme that makes the cut. There's the place on the DNA where the cut is made that's called the restriction site. And then there's the number of fragments created as a result of those scissors being applied to the DNA. Okay, moving on. So the ECAR-R1, they already described that. The enzyme cups right there and creates sticky ends, and you've got that. Okay. Other examples. ECAR-R1 isn't the only enzyme out there. There's also the PST1, the ECAR-RV, the HEY3, and the NOT1, right? There's other enzymes. Remember, enzymes are picky about their active sites. Each one has a different base sequence that it's looking for. Each one's slightly different from the other. Oh, I'm sorry. There was. Did you get that? There was a question on there. I totally forgot that. So this is the answer to that question. CCGG would be, right, the restriction site for HEY3, right? Not a difficult question. You got that. All right, let's keep going. Okay, here we go. This is a good one. Look at the following linear strand of DNA. If HEY3, right, which we just said cuts at CCGG, were mixed with it, how many fragments would be created? Remember, it only cuts where it finds a CCGG. You guys have good eyes. What are you seeing? Again, another good example of a potential multiple choice or FRQ question. Okay. Yes. I see 2 short fragments and one fragment on that. That makes 3 cuts, right? It makes 3 cuts. I'm sorry, 2 cuts? Yes. So let's look at the answer. Yeah, you're right, 2 cuts. I thought there was one there, but there isn't, right? So here's 1 cut, CCGG there, so that makes a really little one. There's another cut right here, so that makes a second sort of short one, and then the last bit is left as the third, or 3 fragments created, right? 2 short ones and 1 long one. Everybody get that? Good. All right, moving on. You guys are good. What if your DNA is circular? If your DNA is circular, and a lot of DNA that comes from bacteria are circular pieces of DNA, then it rather changes the number of fragments that comes from a cut, right? Remember, eukaryotic DNA is going to be linear. So that's going to result in different numbers of fragments per cut than if you have a circle, like a continuous piece of yarn, right? And you apply scissors to it, it just makes it a little different. And on this plasmid, you'll notice that there are places for different enzymes to cut. So there's a Hind 3 cut here. There's an EcoRI here, here, and here. There's 3 places where EcoRI will cut. There's 1 place where BAM-HI will cut. I can't see down there because my thing is blocking it, but, oh, I'm sorry, I'll stop moving. There we go. But there's a BAM3 site on the bottom as well. But if only ECAR-R1 is used, how many pieces are you going to get? How many fragments are you going to get? Okay. Okay. So we have some disagreement. Most people say 3. Okay, let's go back and let's look. So here's 1 cut, right? So there's 1 piece from here all the way to here. That's going to be 1 piece that results from that because there's another cut here, right? And then there's this cut here and this cut here. So there should be 3 fragments. Let me show you. See? If you make red lines where the cuts are, that means everything between the red lines becomes a fragment. So there's 1 fragment, there's 2 fragments, and then there's 3 fragments. Another excellent question, right? Okay, let's keep going. See if you can handle this one. Now, what if it's with all the enzymes are making cuts? Every single enzyme makes a cut. Now, how many fragments will be produced? if BAM3 and EcoR1 and HIND3 are all used, and how many fragments will that produce? That's right, 6. Yes, let me show you. Because every bit between the enzyme cuts will become a fragment. So 1, 2, 3, 4, 5, and 6, right? Okay. All right, what if it's only a double digest? EcoR1 and HIND3. Now, not only how many fragments will be produced, but how long will each one be? So BAM3 is not being used, but the other 2 are. Let's see what you can do with that. Okay, see how are you doing here? Okay, so no, you won't get that many fragments, but you will get fragments of different lengths. Okay, yeah, this one looks right. You'll get, I go back to this. The only one not being used is the BAM3 down here, right? So everything, every other fragment is there. So this one, you'll have one 1650 fragment, one 600, one 500, and one 750. And then the last one will be a combination of these 2 down at the bottom, which irritatingly is being covered by the presentation station, but that's going to be a 1500. So there's 1, 2, 3, 4, 5 fragments. And this one is going to be a combination, a nice long one. Oh, no, you're right. I'm sorry. My eyes aren't very good. No, you're right, Carter. Forgive me. So let's see. 1, 2, 3, and 4. Yeah, so there should be 4 fragments. Do you see that? Everyone good? Yeah, okay. And the presentation station is in the way. So 4 fragments total is the correct answer. 4 fragments, that looks about right there. That's good. And that is, again, another really good, yeah, that's good. 4, very good, yes. 4 fragments. It looks like most of you got it. Excellent. Okay, so remember, I mean, those, if you answer this way, beware, because you only get half credit if it's an essay, okay? Make sure that you put the lengths of the fragments in. If they ask for them, make sure you put them in. BP stands for base pairs, just in case you wonder what BP stands for. Okay, so you'll have 4 fragments that are these many base pairs long, meaning the A's, the T's, the Z's, and the G's. That's a good job, guys. That's good. You just probably took care of a couple points on the AP exam there. Okay. So yes, yeah, there you go. There's my answer, and that's correct. 4. Thanks, Carter, for catching me on that. Okay. Okay. Gel electrophoresis. Here it is. This is how you do it. So first of all, you need to take your DNA sample. If your DNA samples are different, like if you're comparing different people, like a crime scene with 2 suspects or something like that, you take the DNA samples and you mix them with a restriction enzyme that cuts them up into bits, right? So not everybody's DNA is going to be identical. They're going to have restriction sites in different places. That's what sets them apart. That's what would set apart DNA from the crime scene from suspects, right? So now you have DNA samples that consist of mixtures of the DNA fragments of different sizes. And they're in here. You can see my, oh good, you can see my arrow there. So each one of these test tubes has a different DNA sample, and it's already been cut up into different lengths of DNA. Then you inject each sample, this is the really cool part that I wish you guys could do, with those micropipettes into these little holes in like this hard jello stuff that I'll let you guys see in a minute, okay? And you put them in these holes, and then the gel is immersed in this bath with a buffer in it. And the gel is polarized. You put an electrical current through it so that the black side is negatively charged, right? And the red side is positively charged. Now DNA, what do you think? Is DNA going to be more negatively charged or positively charged? What do you think? It does have an overall charge. It is negative because of all those phosphate groups with all those oxygens on the sides, lots of electrons on those oxygens. So very negative. So if you start the DNA over here and you put an electric charge so that this side is positive, it draws the molecules towards the positive end. And that's how it works. Okay. So here's a question. DNA samples are injected into the wells at the blank end of the gel. Right? Nice picture. Okay. This DNA is a negatively charged molecule, and you start it on which end is the question. Okay. At the negative end, yes, you start at the negative end. You see how the wells, the holes in the gel are here, and this is where you're putting your DNA samples at the negative end of the gel. And then when you turn the current on, they migrate towards the positive end, which is down here. Nice job. Okay. When you turn the power on, DNA fragments move towards. Would you like me to put these on the test? Shall I make these multiple choice questions for you? Mm-hmm. Would that be nice? Just give you a bunch of questions with only 2 choices each. Those are always nice. Yes, towards the positive end. Beautiful. So they start at the negative end, they move towards the positive end. Yes. The relationship between fragment size and distance traveled on the gel. Take a guess at this. So the larger the fragment size, the more or less distance you think they will travel. It is an indirect relationship. Yes, it's indirect, meaning the larger the fragment, the shorter the distance it's going to go. That should make sense, right? That the larger the fragment, the more difficulty it's going to have making its way through the agarose gel, and that's exactly what happens. And that allows the shorter fragments to go further and the longer ones to not go as far, and that creates the pattern that allows you to do some analysis. So it's an indirect relationship. The longer fragments go less far. The larger the DNA fragment, the less distance it travels. Okay, then it's time to analyze the data. So then you get this ridiculous acronym, RFLP, which stands for Restriction, Fragment, Length, Polymorphisms, which is just a fancy way of saying the pattern of bands, right, show that you're comparing on your DNA electrophoresis gel. Look different, because the fragments don't travel as far when they're long and they travel further when they're short. That's it. You're just looking at the patterns on the gel, and that's what RFLP stands for. Okay, fingerprinting. All right. In humans, you can actually take a person's DNA sample and create a fingerprint from it. So, for example, this is taken right off an AP exam question. Looking at the hemoglobin gene. This is the gene for normal hemoglobin that makes good hemoglobin so you don't get sickle cell anemia. And this is the one for the mutant that has a mutation right there. Right there. And then when you electrophoresis this piece versus this piece, after you've cut it with a restriction enzyme, you get these patterns. Now what you need to know is this hole here and here, these are the holes where you put the DNA. They're not part of the pattern, okay? These are part of the pattern for the normal. This is the banding pattern for the sickle cell. Okay, going. Yeah, not a difficult question. What's the name of the restriction enzyme? What do you think it's called that's doing this? Mm-hmm. Right. Okay. Yeah, that's it. That's all. You just had to identify from the picture what the name of the restriction enzyme was, and you got it. DDE1 is the name of the restriction enzyme making the cuts, okay? And the normal beta globin, this one, was cut into blank fragments, and the sickle cell was cut into blank fragments. Don't worry about the tiny bits on the end there. Okay, just ignore that. Just use the patterns here. Give you a better answer, right? Excluding the holes at the top. The wells at the top are not part of the pattern. So here's normal, here's sickle cell. Right. So the normal one is 3 and the abnormal one is 2. So it's 3 and 2. There, 3 and 2. Okay. So there's 1, 2, 3 fragments from the normal, and there's only 2 for the mutant because of that mutation right there. Ruins the restriction site that the DDE is looking for. I just gave the answer, but oh well, that's fine. Okay. Yep. Because one of its restriction sites mutated and the enzyme no longer recognizes it as a place to cut. That's it. That is it. The restriction site that should be here got mutated. And if you change the DNA sequence, you change the ability of the enzyme to recognize it as a restriction site. Good. Did that. Okay. Forensic evidence and genetic profiles. All right. So we talked about this. Short, I'm not going to worry about that. Okay. Okay, so this is a really interesting example. Earl Washington was 17 years in prison, and it took a while for them to get DNA fingerprinting into a legitimate science, something that could be recognized in a courtroom. So what happened with him was that when they went back and they took the DNA samples from the crime scene, he was accused of rape, and they compared his data, here's Earl, right? And here are the markers in Earl's DNA compared to the markers of the DNA on the victim, look at that, versus Kenneth Tinsley, look at him, right? It looks like he's a perfect match for the markers that they were looking for in the DNA that was found in the crime scene. So you can see that Earl Washington was exonerated. It couldn't have been him because the technology was sound enough for them to be able to trust it in the courtroom. So it wound up exonerating him, and he was actually released. Another good example, if they show like an image of a crime scene DNA here, and then on the right, they ask you to tell which suspect you think was most likely at the crime scene, was most likely culpable. So what would you say with this one? It looks like suspect 2 is a perfect match for the crime scene. The banding pattern on the gel electrophoresis shows a very similar pattern. Okay. All right, so I just want to show you to the sperm DNA here, okay? This is, again, it's a sexual assault case. This is the crime scene DNA right there. And then suspect 1 and 2 are here. And then the boyfriend's DNA fingerprint is there, okay? So you're comparing to the sperm DNA. So who most likely did it, given that information? Here's the sperm DNA, suspect 1, suspect 2, and the boyfriend. And that's an real data. That's a real gel. So, yes, suspect 1, definitely. Yes. Look at suspect 1. Look at those 2 bands. Exactly match the sperm DNA that was found on the victim, right? Looks like the boyfriend is exonerated. Their bars are way up here, and suspect 2 just has that 1 marker there that isn't anywhere near what was found at the scene. Okay, see how we did that? Okay, excellent. Almost done. 1. Okay. Okay, that is it. All right, so I actually don't have a